In classical evolutionary genetics, sustained directional selection operates like a one-way ratchet. The standard axiom assumes that if you expose a population to intense, constant environmental pressure over thousands of generations, it will eventually exhaust its standing genetic variation. This process is often modeled as a hard sweep. A highly beneficial genetic mutation appears, and because it provides a survival advantage, it rapidly rises to 100% frequency across the population, purging all linked variation in the surrounding genome along the way. The mathematical consequence of this sweep is a rigid endpoint. A population eventually reaches a mutation selection drift equilibrium, a state where all of the older, less advantageous genetic variations are permanently lost. But biological reality is rarely as clean as classical theory. It remains entirely unclear whether this model actually holds true for complex, polygenic life history traits in large, sexually reproducing populations. If the hard sweep model is an absolute rule, taking a population that has undergone 1,000 generations of extreme specialization and throwing it back into its ancestral environment should result in a mutation-limited evolutionary dead end. To test this, a 2026 study led by Kenneth Arnold and Mark Phillips used Drosophila melanogaster in an experiment designed to push the hard sweep model to its breaking point. The researchers maintained two radically different experimental regimes. The A types were selected for rapid early reproduction on a strict 10-day cycle, while the C types were selected for delayed reproduction on a much longer 28-day cycle. The A-type populations provided the ultimate stress test. They had been subjected to intense, constant directional selection for early life history for over 1,000 generations. This chart tracks the genome-wide expected heterozygosity of these flies. The blue box plot on the far left at generation 0 shows the toll of 1,000 generations of selection. By all standard sequencing metrics, this genome is classically exhausted. The genetic variation has been entirely purged. With this baseline established, the researchers triggered a reciprocal shift. They took long-standing A-type populations, forcing them into the delayed reproduction C-type environment, creating an A to C trajectory, while forcing C-types into the rapid A-type environment. If A-types truly lost their genetic variation to fixation, their biology is now a closed system. Forced into a delayed reproduction regime, they should be completely unable to adapt, resulting in a catastrophic physical and genetic failure. Specializing for 1,000 generations creates an enormous biological hurdle for reversal. Without standing variation, waiting for new mutations to arise and spread should take tens of thousands of years. Yet the adult mortality assays immediately following the regime swap showed a nearly immediate response. These graphs plot differences in age-specific mortality between the evolving populations and target regimes. The dark blue line in the lower graph represents the A to C trajectory at 51 generations. It has rapidly flattened across the lifespan, converging cleanly toward the delayed reproduction baseline. To corroborate this, the researchers analyzed a second, entirely distinct phenotype, developmental pupariation timing. This chart tracks the terminal results for developmental timing, showing a clear clustering of trajectory lines. The green A to C trajectory abandoned its ancestral early reproduction trait, the red lines. It converged perfectly on the late pupariation C-type phenotype, represented by the blue lines. By regressing to their precise ancestral states within a few dozen generations, these populations demonstrated that their evolutionary potential remained intact, despite a millennium of specialized selection. Random mutations cannot explain this. A mutation-driven process wouldn't produce such a rapid, identical, and coordinated phenotypic shift across 10 independent replicate populations. The genomic data reveals a highly polygenic architecture at work. The adaptation was distributed across nearly 600,000 distinct single nucleotide polymorphisms, or SNPs. This principal component analysis charts the global genomic trajectory of the populations over time. The replicate populations followed crossing paths. The blue lines for the A to C populations and the red lines for the C to A populations moved in precise opposing directions across the principal component space. To understand how this global reversal is possible, We have to look locust by locust to see if the exact same alleles are driving both directions of the shift. This hexbin density plot maps allele frequency changes for all 600,000 SNPs. The density concentrations sit strictly within the second and fourth quadrants. This diagonal alignment proves an anti-parallel shift. Alleles that increased in frequency under one regime decreased in corresponding proportion under the other. This shift presents a mechanical contradiction. 600,000 specific alleles cannot move in a coordinated anti-parallel dance if the A-type founders had already purged their genetic variation at those exact sites. The genome isn't blindly guessing with new mutations. It is walking backward along a pre-existing but seemingly invisible polygenic path. To find that path, we have to look for the hidden data that standard low-resolution sequencing methods completely miss. The A-type genomes that supposedly lost their heterozygosity to 1,000 generations of directional selection still fueled a massive polygenic adaptation. When we track the genome-wide heterozygosity of the A to C populations after the selection is reversed, we see a striking progression. There is a rapid and progressive vertical climb in heterozygosity across 65 generations. Under the rules of classical genetics, completely lost alleles cannot resurrect themselves. To investigate, the researchers ran deep sequencing on the founder populations at 767x coverage, allowing them to look past the limits of standard 90x sequencing. This histogram tracks the frequencies of those supposedly vanished minor alleles through that high-resolution lens. The purple bars show that the sites that previously appeared completely fixed actually harbored a hidden reservoir of ultra-rare alleles, sitting at a mean frequency of just 1.6%. In complex traits, fixation is often an illusion created by low-resolution sequencing. The genetic variation was never exhausted, it was actively hiding. Under the drift dynamics of a population size of 900, a minority allele at 1.6% faces a high probability of loss over 1,000 generations. In a neutral system, random chance would have eliminated these variants long ago. Yet, these alleles survived across 10 independent replicate populations. This repeatability demonstrates they weren't floating randomly, they were protected by an active selective force. This points to a genomic architecture known as antagonistic pleiotropy, a form of balancing selection. In life history traits, antagonistic pleiotropy creates a trade-off. An early reproduction advantage imposes harsh penalties on late life survival. Because no genotype achieves perfect fitness across the lifespan, this acts as a genetic anchor. It prevents true 100% fixation, shielding the minority allele from drift. This architecture ensures a genome never burns its bridges. Even under extreme selective pressure, it archives its ancient blueprints at cryptic frequencies. Sustained directional selection does not result in an evolutionary dead end. Through balancing selection, the genome maintains a cryptic reservoir of standing variation, ensuring that populations retain the architecture required for ultimate reversibility.